Thank you. Yeah, this research was done in my undergraduate very recently, so very new to the field, and it was on the mutations in the Savannah monitor lizards, Nicotinic acetylcholine receptor conferring reduced susceptibility to Sympatric Cobra venoms. So jumping straight in, alpha-neurotoxic venoms have evolved to target nicotinic receptors. This causes flaccid paralysis in their prey, and toxins such as the 3-phenyltoxin have varying degrees of affinity to specific amino acids along that binding site that they're targeting. So, as Kevin said, co-evolutionary arms races cause a lot of diversification, and in terms of the neurotoxins, this has caused key positions along the receptors of prey species to reduce sensitivity to alpha-neurotoxins. In the case of the mongoose there, it's N-glycosylation of asparagine residues, which causes steric hindrance, which physically blocks the toxin from binding. Of note is position 191 and 195, which guides the toxin to bind to the receptor. So what I did is I looked at Australian Varanids and Varanus exanthematicus, which is an African monitor lizard. And at position 191 and 195, it's got a glycine and a asparagine, which are both uncharged residues. Whereas the Australian-derived Varanids have a single negatively charged amino acid at position 191 in the case of Varanus giganteus and Varanus komodoensis, or 195 in the case of Varanus mertensi. This gave us a good testable hypothesis that removal of charge at these sites will reduce the binding of alpha-neurotoxins. In addition, we also had a mutant, which had two negatively charged aspartic acids in place 191 and 195 of the receptor. And this is what we found. So very broadly speaking, we've got Varanus komodoensis there in green with Varanus giganteus, as well as Varanus mertensi in the blue there. Across all neurotoxic species, they were susceptible to venoms, so very strong binding seen there. In addition, the mutant, which is in very dark blue there, did not really increase sensitivity. So it's key that there's just one negatively charged amino acid there for successful binding. And finally, we've got Varanus exanthematicus there in the red, and across all neurotoxic venoms, there was reduced susceptibility. So by removing the charge at these key sites, we've conferred reduced susceptibility, and the toxin can't bind as well to the receptor. So understanding why this might have occurred in exanthematicus, you have to take a look at the ecology. They're quite a small-sized monitor with a really high diversity of large neurotoxic predators. There's a lot of cobras living sympatrically with them in Africa. So they're under a really high selection pressure. In the case of the Australian-derived Varanids, they're all really large species, the ones that we tested, making them less vulnerable to predation without the presence of these really large cobras. Something like a death adder is not going to take the Komodo dragon because of just the pure size of it. This implies that the reduced susceptibility trait is either use it or lose it. Again, hammering home the large neurotoxic predators needed to maintain that selection pressure on the lizards. And with any evolutionary novel trait, there's possibly a trade-off to do with having that trait, and that could possibly be, with a bit of speculation, the reduced efficacy of the acetylcholine molecule. It might not be able to bind as well with reduced susceptibility, so they could have subsequently lost it once they radiated away from large neurotoxic predators. Or it might just be the case that combined with thick scales, reduced susceptibility does the trick and they're able to escape. What you can see in the picture here is a close relative of exanthematicus. This is Varanus albigularis, and it's trying to be eaten there by a large cobra. I think it's Naya annulifera. I'm sure somebody in the audience would be able to correct me on that one, but this was in Kruger National Park, and it was something like a 15-minute long battle where the cobra was trying to eat the lizard and it was just tanking its way through. Fortunately for the lizard, a kind tourist went and scared the snake away. The lizard laid there and then eventually just walked off. So leading us to speculate that albigularis may also have reduced susceptibility to neurotoxic venoms, but further testing of Afro-Asian varanids combined with these natural history observations are really key to understanding the resistance traits. Thank you very much. It's a short and sweet talk, and thanks for listening. Big thanks to Brian Fry and Richard Harris for supporting me along. Yeah, that's it.